Okay.
Okay, thank you all for being here today. This is like a very bittersweet moment for me because this is my second graduate student, or second PhD student who is finishing. And I wanted to tell you a little bit about Omar. First of all, he got his Bachelor in Science in Pharmaceutical Sciences from UC Santa Barbara. He was an undergraduate researcher in the Metrology lab, studying formulation of drug conjugates for oncology. He also interned at the California Nanosystems Institute, where he was a lab assistant doing next generation sequencing. Following graduation, he was a research associate at SB Bio in Santa Barbara. And that's where he learned a lot of immunochemistry, did histology, QC, research and development. That brought him to SDSU. So Omar reached out to me. I was my first year, 2018, 2019. I just started. My lab was kind of still in boxes a bit, but he was a prospective graduate student wanting to apply to the program, but looking ahead at what kind of labs he would be interested in. So very proactive. He came to me and said, Dr. House, I really want to study fibroblasts and chemoresistance and relapse. I really want to understand how chemotherapy affects fibroblasts. And I thought, okay, this is just for fibroblasts is not my expertise. I've really had very little experience in the tumor microenvironment. But I could tell based on his engagement and his knowledge and his enthusiasm that he would be a great student. And I went home to my husband. I said, I don't know what to do. This student wants to join my lab, but I have no fibroblast experience, but he really wants to study fibroblasts. He's like, he's a really great student. It'll be fine. That's all that matters. And as it turns out, Omar's been a fantastic student. He really drove the project that he's going to talk about today. Interestingly enough, about 6 months into his project, he got completely scooped, and he'll talk about that paper a little bit today. But he was able to take that information and build on it, what other people have published. And that's like one of the best things you can hope for in your graduate student is that they take data from the field and build on it and develop a project out of it. And he also, which you'll see, kind of made connections from other projects in the lab to build this, really cool interesting work that he did. So this is us in the beginning when he first, around the time he first joined the lab. Everybody's happy. This is like 2019. And then we get to, oh, it's not going to go. Then we get to the pandemic. Omar is still pretty happy. I know you can't tell, but he's actually smiling under there. He was very steadfast in maintaining the progress of the lab and also of his project. This is post-pandemic. We're celebrating as a lab, happy again. Everyone's, excited. Omar then went several times. He visited AACR and presented his work. This is again post-pandemic. Everyone's happy, going to New Orleans, having fun, going to AACR. I also wanted to highlight again that Omar presented at S Cube. This is our annual research symposium every year. And he won the Dean's Award, I think, this year, that year. Yeah, so he won the Dean's Award. And he also, I'm not going to go over all the awards he won, but several of them for his presentations, and even at the Interdisciplinary Cancer Research Meeting, and then also, again, at S Cubed. Some other awards he's won is the Reese Steely Research Foundation Fellowship, the Core Fellowship, as well as the SDSU UGF University Graduate Fellowship. And so I want to emphasize again that Omar has been a fantastic student. He came through the pandemic shining, ready to go, always enthusiastic, very engaged in the work. It can get really, as most grad students know, it can get pretty hard to get through this, to get through grad school. And it's very difficult. And he really maintained a lot of enthusiasm and engagement. He contributed to lots of different projects in the lab. He's co-author on several manuscripts that come out of our lab. in lab meeting, he's always, offering his input, his comments, and his advice. He created and he developed the IHC core that we have in our lab, so we're able to, process tumor tissues and do histology, et cetera. He developed that in our lab and optimized all the protocols for it. And he trained many students, several undergraduates who are now in grad school or in industry doing fantastic work. And so I just want to highlight fantastic student, and I'm really proud of the work that he's accomplished and how much he has driven and taken ownership of the project, and hopefully that will come through in the presentation today. And so this was us at graduation, and I'm really proud of you, Omar, and we look forward to hearing about your work, and congratulations.
All right. Well, thank you everyone for being here. I appreciate it. And I will go ahead and get to the science. So the title of my talk today will be Alternative NF-kappa B signaling promotes AV beta 3 mediated stress tolerance and relapse in high-grade serous ovarian cancer. So this presentation will be broken up into 4 pieces: background, hypothesis, specific aims, and conclusion. To get started with background, ovarian cancer is the most common cause of gynecological mortality in the US, with an estimated 14,000 deaths this year. And within ovarian cancer, there's 3 histological types, with the epithelial type being the most common. And within this epithelial type, we also have 4 epithelial subtypes. And the most dominant here being serous. Within serous, there's high grade and low serous. High grade serous being more common and also more aggressive than the low serous form. And in terms of high-grade serous ovarian cancer prognosis, consist of 4 stages. The first 2 stages is not as common in terms of diagnosis, with the stage 3 and 4th being the most common stage of diagnosis. This is due to a lot of the symptoms are very negligible in terms of other potential things that occur, such as bloating and things like that. And in terms of the 5-year survival rate, if it's caught in the early stages, it's around an 80% chance of survival. But where it's usually commonly diagnosed, the 5-year survival rate's closer to 15%. And so in these later stages, what occurs is we have this formation of this fluid called the ascites fluid, and this fluid fills up the peritoneal cavity. And what this allows for is for some of the cancer cells from the primary site to slosh off and float into this environment. And when that occurs, it allows for a type of cancer cells that are essentially floating around in this space and forming spheroids. So these small metastatic lesions essentially begin to float around in this fluid. And this leads to an interesting form of metastasis for this cancer, where it's transcolomic metastasis, meaning that it's spreading through the peritoneal cavity compared to what others may be more commonly known for in terms of like other cancers like breast cancer, where it might be more through the bloodstream or lymph nodes. And so, as I mentioned, in the later stages is when this is usually diagnosed. Then the primary site of metastasis is the omentum, which is this fat pad that covers the abdominal wall. I know some of you guys are eating, so this might get a little bit graphic, but ultimately, here just showing that it really likes the omentum and it completely fills up this cavity. Usually, debulking surgery needs to happen. So, this whole tissue, this whole organ gets removed from the body and given chemotherapy. And over 80% of patients will still relapse within 2 years, even with chemotherapy and complete debulking surgery and removal of this omentum. And what we think is happening is these tumors are very heterogeneous. And here we have them split into what we call the bulk of the tumor cells and this smaller portion of cancer cells called cancer stem-like cells. And so the bulk of the tumor cells that you can see here are in the dark pink here. After surgery and chemotherapy, those cells are quickly capable of dying with chemotherapy and the debulking surgery. But with this cancer stem-like population down here is considered more quiescent, so it's not as proliferative in terms of the cells there. It's also, they're very chemoresistant, and they have attributes to normal stem cell biology or normal stem cells. And so how I would like you all to think about these cancer stem-like cells is more of when you think of a plant, you have the top of the plant that you get to see, and then you have the roots. If you try to get rid of a weed and you just plop the top of it, the roots are still there. So because the roots are still there, the plant can regrow again. And so you can think of these cancer stem-like cells as those roots there. In terms of the molecular biology, the way that we identify these cells is with multiple cell membrane markers as well as transcription factors and some enzyme activity. For this presentation, I'll be focusing on the use of these two markers here, CD117 and to some extent CD133. And these markers have been extensively used in our lab, and a lot of our models and phenotypes have been used through the identification and isolation of these cancer stem-like cells. So ultimately, what we're trying to figure out is what mechanisms promote and or maintain these cancer stem-like cells during chemotherapy exposure and relapse. And one way that we know that high-grade serous ovarian cancer progresses is through NF-kappa B transcription factors. And when this pathway can be broken up into two main signaling arms. And I know it's a little complicated here, but ultimately, when I say canonical, this is talking about the classical pathway, and it relies heavily on RelA. So when I say RelA or classical pathway, we're talking about this signaling arm here, where stimuli such as TNF alpha, which is a classical activator of the classical pathway, activates and recruits cofactors that then leads to the ubiquitination of the relay complex. This essentially degrades parts of this protein that allows for this relay protein to get into the nucleus and essentially become activate and do its job. When it comes to the non-canonical pathway, or also known as the alternative pathway, this is driven through the RelB transcription factor. And this pathway is activated by a separate set of stimuli. Here showing tweak as one of these. Tweak is tumor necrosis factor weak inducer of apoptosis, and it binds to this receptor here. We see it's called F14 and leads to activation of RelB by ubiquitination of this P100 subunit, leading to it being translocated into the nucleus here. And ultimately, what this means is, in terms of the research done, when you think of Relay's programs are more involved in proliferation, while with RelB seems to be, like I mentioned before, more like quiescence and chemoresistance. The way that we've figured out some of these mechanisms here is when you put cancer cells in ultra low attachment conditions. So this is supposed to simulate more what you see in that environment where the cancer cells are in that peritoneal ascites fluid. And so these cells, since they're not allowed to sit down and attach to the bottom of the plate, they rely heavily on the RelB transcription factor. Well, when we put cells in monolayer conditions, they don't really use it as much and rely more on RelA transcription factor. In terms of these transcription factors as well, in an orthotopic model, so this is when we put cancer cells in the region of the mouse that is close to the ovaries, and we use small hairpin RNAs to either knock down RelA and RelB. And what we find is that when you either reduce RelA or RelB, we lose the ability for these cancer cells to metastasize to different organs. So with this, we also were interested in seeing the different type of stimuli that can activate this pathway. So a previous graduate student looked into an array of cytokines, some that are classically already established in terms of activating NF-kappa B factors. What we see here is in terms of tweak, again, being tumor necrosis factor weak inducer of apoptosis. I know it's a very long acronym. Tweak activates NF-kappa B pathway to a similar manner as a classical activator like TNF alpha. When we look deeper into patient samples before and after treatment, what we see is this upregulation of this or enrichment of this tweak factor in the post-chemotherapy setting. And when we bring this back to the idea of our cancer stem-like cells, here looking at marker CD117 positive cells, when we put them in conditions such as either with tweak exposure, carboplatin, or in combination, we began to see this enrichment of this vector. In terms of this publication, we also looked at phenotypes behind this and found phenotypes that are associated with these cancer stem-like cells and tumor-initiating cells. So going back to this 2D, 3D culture system that I was talking about with the 3D spheroids, allowing for a phenotypic showing of these cancer cells in the peritoneal space, what we did is we did some RNA-seq, and we also induced knockdown of REL-A through small hairpin RNAs, and these are inducible. And so this next graph is a little intense, but I will break it down a little bit. Ultimately, this is gene ontology of the top 20 hits or so that in the 2D and 3D conditions with either knockdown of REL-A or REL-B in either 2D and 3D. And there's many things that these two pathways are regulating in a different manner. But I'd like to point out that a lot of these have to do with adhesion, either to cell-to-cell, cell to a substrate adhesion, extracellular matrix organization, focal adhesion, integrin 1 pathway, and positive regulation of cell adhesion as well. So with this in mind, so a lot of these adhesion-mediated mechanisms is involved through the use of these membrane receptors called integrins, and integrins come together as heterodimers of an alpha and beta subunit. And when they come together, they attach to ECM or a substrate that is around the environment. And when they attach to the extracellular matrix, they have different phenotypes in cancer that are involved in proliferation and chemoresistance. And in terms of these heterodimers, they have different affinity for different substrates. So for example, an alpha 5 beta 1 subunit will come together and attach readily to fibronectin over something like collagen. So using this RNA-seq data, we wanted to go deeper into these integrins and see if REL-A and REL-B are differentially regulating some of these subunits, and also looking at conditions of 2D and 3D. And what we found was that in this knockdown of REL-B in this 3D condition, we find that ITGB3 is specifically being downregulated in 3D conditions when we knock down REL-B only. And in terms of in terms of ITGB3, beta 3's most known partner is alpha V. And when we look at ITGAV, which is up here, we see that that's being regulated mode through the knockdown of RLA in these 3D spherical conditions. So with all of this, we hypothesized that NF-kappa V mediated integrin expression enables survival of CSCs to a chemotherapy modified environment to facilitate tumor repopulation and relapse. And there will be three aims that I'm going to be highlighting in this presentation. Aim one being characterizing the role of REL-A and REL-B and in regulating integrin expression in these ovarian CSCs or cancer stem-like cells. And this will cover relationship between tweak, NF-kappa B, integrin expression, and some relapse properties that I will show. And aim two will be evaluating the contribution of chemotherapy-altered tumor environments to AV beta 3 mediated metastatic growth and relapse of these ovarian cancer stem-like cells. And with this aim, we're trying to highlight how fibroblast-derived extracellular matrix following chemotherapy can mediate tweak, FN14, and NF-kappa B dependent cancer stem-like cell adhesion and survival within an in vitro, ex vivo, and in vivo models. And finally, for aim three, we're investigating the potential therapeutic benefit of inhibiting REL-B-dependent AV beta 3 signaling in combination with integrin antagonists to prolong survival and relapse. And this is to see the feasibility of targeting this cascade and how this may potentially reduce these cancer stem-like cells to ultimately increase the survival rate for these patients, or at least in these models. And so for aim one, I will be highlighting some experiments in terms of the techniques that we'll be using here is flow cytometry. So here is to assay the these integrins on the cell surface, since that's where they functionally are. We'll also be doing some magnetic cell sorting. This allows us to separate our cancer stem-like cells from the rest of the tumor cells, and some in vivo models as well. So to start off, I showed you this RNA-seq data. We see that there's this regulation between these two subunits, so we wanted to first and foremost validate this with actual flow cytometry and see if these integrins really are being upregulated in the 3D condition, right, in these spheroids, and also is RelA or RelB involved. As here, we would predict that with knockdown of RelB, we should see a loss of Aβ3 expression on the cell surface. And what we find is when we do flow cytometry and we look at the percent of Aβ3 and we're looking at 2D and 3D conditions, we began to see that in 2D, where there's really no changes, and especially when we modulate by knocking down either RelA and RelB, here shown as SHRelA or SHRelB. But when we look at the 3D conditions, we do see this significant enrichment of Aβ3. And when we knock down either RelA or RelB, we begin to see this loss of expression of Aβ3. You might be wondering, Omar, when you knock down RelA, you're also seeing this. Well, RelA is a transcriptional regulator of RelB expression. So when we knock down RelA, we also lose some RelB expression. However, when we knock down RelB, we do not lose RelA expression. When we go ahead and do this also in terms of chemotherapy and more stressful conditions, so adding chemotherapy to the setting begins to enrich some of these Aβ3 integrins on the surface. 3D conditions, again, is also modulating this expression, but when we combine these 3D spheroid conditions and chemotherapy, we really see this enrichment of Aβ3. So to really look deeper into this, we wanted to do a relapse model, an in vivo relapse model, where we took OVCAR-A RFP luciferase cells. We took 150,000 of these cells, and we injected it intraperitoneally into the peritoneal cavity of mice. And after this injection, a cohort of these mice were either given vehicle, and another cohort was given chemotherapy, carboplatin and Taxol, which is usually one of the combinations used in the clinic. And throughout the progression of these tumors, we image using IVUS imaging. And after endpoint criteria, we assayed with flow cytometry, looking at Aβ3, Aβ5, and beta-1 active. These integrins are implicated in the post-chemotherapy setting. So this is a reason why we wanted to look at this. And also, of course, the previous data that I was showing. So what happens in this model? Here I'm showing you luminescent counts. So these are the counts from this IVUS imaging. In the black here, these are the mice that were given the cancer cells, but no challenge of chemotherapy. So as you can see, these over time will continue to progress and develop higher tumor genesis. When you give them 3 rounds of chemotherapy in the other cohort, you quickly begin to see that the tumor load drops. This dotted line here is representative of our background mice control. So as you can see, for a couple of days or so, we essentially lose signal. And after some time, these tumors start to come back and relapse. And at day 68, where we find ourselves back at the start of where we first injected these cells, we want to go ahead and took the tumors from this endpoint here. And we then assayed for integrins as well as where the cancer cells are going in different organs. For now, I'm just going to show you some of what happens with the integrin story. So we can see is in this chemotherapy setting, the cells that we grabbed from this group here, they have a high enrichment of Aβ3. Our vehicle mice have expression as well, just not to the same amount. And in terms of Aβ5, we also see that there's this increased expression of Aβ5, just not to the same magnitude as Aβ3. Beta-1 active integrins were also assayed here, and we see this upward trend, but due to the variance, we don't see significance. And now to bring this back to the story with, yes.
So in your controls,
Mhm.
you're up at 68 days. How much tumor necrosis was happening in the tumor cells there? There's a lot of stuff going on, a lot of inflammation.
Yes.
A lot of survival. So you doing live dead sorting of your
Yes. Yes. And also specifically, these cancer cells have RFP expression, so that's another way that we're assaying specifically for the RFP.
You're still
Yes. Yes.
Thanks.
Yeah. So now bring this back to the story of tweak. Um, we've previously published that when tweak binds to FN14, its only known receptor, it activates the RelB pathway and leads to post-chemotherapy relapse in ovarian cancer progression. And so the question here is, are NF-kappa B transcription factors, RelA and RelB, involved in this AV beta 3 regulation, and specifically in these cancer stem-like cells? So the way we did this, we again looked at flow cytometry, and we looked at the expression of AV beta 3 in our either CD117 negative, which is this lighter orange color here, and our CD117 positive cells, which is this darker pink salmon color. And off the bat, what we can see is compared to our negative cells, we have very low expression of AV beta 3. But in our cancer stem-like cells, they do seem to have this enrichment of AV beta 3. And in the CSCs, when we knock down RelA and even more so RelB, we begin to lose this expression of AV beta 3. And when we add tweak, we do see an induction of AV beta 3 only in these cancer stem-like cells. And when we knock down either RelA and RelB, we begin to also lose this effect significantly. And we can see this in multiple cell lines. And we also assayed other integrins such as alpha 5 beta 1. And what we found here is that these heterodimers are highly expressed in either our CD117 negative or our CD117 positive cells. And when we go back to this relapse model and we specifically ask the question of what are our cancer stem-like cells expressing, and we looked at our expression of either AV beta 3 or AV beta 5. AV beta 5 is important here because there's literature showing that when even in the presence of AV beta 3, you may have some compensatory mechanisms. And the literature shows that AV beta 5 may be important in that process there. And when we look at just our cancer stem-like cells in these endpoint measurements here, and we look at specifically at the CD117 expression of our RFP positive cells, which are only our cancer cells, we see this switch from AV beta 3 positive, AV beta 5 negative to a predominantly AV beta 3 positive, AV beta 5 positive population here. And another thing that we did with these tumors is, so they have RFP and they also have luciferase. So at endpoint measurements, we took the tumors and we dissected each organ separately and then did a luminescent readings on that's organ specific. And from this, we can then go ahead and take the total luminescence from the tumors of one mouse and then allocate how much luminescence readings we get from, say, the omentum versus the mesentery. So this, when we do this type of a measurement, what we saw was that in the mesentery, after chemotherapy and once those tumors came back, we saw this predilection for mesentery growth post-chemotherapy there. And in a different model, we've also seen this. So in the model to the right, it's with the OVCAR8 cells that I was talking about. In the left, this is a COV4 cell line, and we did an IP injection. And here is with 2 million cells. And in this model here, we assayed with looking at weights. And what we can see again is that in the mesentery, after chemotherapy, and once these tumors regrow, we see that they have this significant increase of preference to grow in this site. So AIM-1 findings, the Go analysis reveals that RelA and RelB differentially regulate distinct pathways and integrin subunits. RelB specifically drives beta 3 expression in 3D spheric conditions and CSCs, and tweak further induces this, as shown by that flow cytometry data. In our relapse model, we see that AV beta 3 and to a lesser extent AV beta 5 are enriched in relapse tumors and CSCs. And post-chemotherapy, tumor burden shifts from the omentum to the mesentery. So the relevance in the field here, there's been publications showing that in beta 3 positive tumors, either having already high beta 3 off the bat or being induced, and we do NF-kappa B pan inhibitors. So this is just inhibiting the universal both sides of the pathway, knocking down NF-kappa B with these inhibitors in these beta 3 positive cells causes for drug sensitivity in these tumors. However, what we found is that even within the NF-kappa B pathway, RelA and RelB are differentially regulating this beta 3. And what we see is that RelB here is predominantly in the regulation of beta 3, while RelA seems to be more involved in the upregulation of alpha V. And in terms of the cancer stem-like cells, we see that our CD117 positive cells predominantly are enriched for this AV beta 3. And AV beta 3 has been extensively published and researched in many tumor models, and they are critical mediators of stress response, as shown here. When there's stressors such as isolation stress, they use AV beta 3 and an array of other genes to begin to generate an ability to not be so stressed out and essentially giving these stress tolerant cells. And in our models, we've also seen similar things when we do an in vivo model and we look at early tumor development versus late tumor development. We see that beta 3 is enriched early on, and then after tumors are predominantly developed in there, beta 3 is no longer used here. We've also seen this with nutrient stress, and these are a set of experiments done by a previous PhD student. And what we can see when we inhibit insulin growth factor receptor, we begin to see this induction of AV beta 3 expression in multiple cell lines. So for AIM-2 now, want to evaluate the contribution of chemotherapy-altered tumor environments to AV beta 3 mediated metastatic growth and relapse of ovarian CSCs, so cancer stem-like cells. And here is, we know that chemotherapy does a great job of getting rid of the bulk of the tumor. And here we're really trying to understand the type of environments that these cancer stem-like cells like to preferentially grow at and be at after chemotherapy. And this would allow us to intervene there so that we continue to use a standard of care, but also understand that the therapy itself and also these cancer stem-like cells will preferentially find ways to evade and come back. So, as Carrie mentioned before in the beginning of this, I was interested in fibroblasts and what they may be potentially doing in these processes. And this came from the work that I was doing in my old company, where I was looking like constantly at slides of different tumor types, and we'll always see these little cells around these tumor cells. And what we can see here is in brown, we're looking at the collagen and fibronectin expressed in these tumors, and these ECM components are predominantly being driven by cancer-associated fibroblasts, and these fibroblasts are the main secretors of this. And you can see it in pre-chemo, fibronectin here in brown, and as well as collagen here being surrounded by these cancer cells. And what this paper showed is that post-chemotherapy of matched samples at either the omental site here, we began to see that the cancer cells that are left over are being encapsulated by this array of ECM, including fibronectin again or collagen. And when we look at the mesentery and the mesenteric metastasis after chemotherapy, we see a similar thing where collagen and in fibronectin, we see that these cancer cells survive and they are being encapsulated by this extracellular matrix. And this study really began to show that this coupled mechanism of cancer cells and extracellular matrix and how in together they help each other in terms of these cancer cells survival after chemotherapy. And so what we want to do here is potentially we know that these cancer-associated fibroblasts, specifically the MyCAF population, are the extracellular matrix generators. And so we wonder if what we're seeing here in terms of our preferred tumor growth after chemotherapy in the mesentery is this extracellular matrix important in that setting there. And so to investigate that further, we generated a chemotherapy activated fibroblast cell-derived matrix system to assay this in vitro. We also did some ex vivo models to look at this and also an in vivo model. And due to time, I'm going to focus mostly on this FCDM system and predominantly in this in vivo model we did. So first and foremost, like I mentioned, that other publication really looked at cancer cells and chemotherapy and the extracellular matrix. And here we wanted to first just ask, like, if chemotherapy itself modulating the environment in a way to induce expression of these extracellular matrix components. So what we did is we took mice and we gave them either vehicle or chemo without the presence of cancer cells. And we divided the groups based on the region of the body where they're at. So in terms of the omentum in the mouse cavity, it sits closer up towards the diaphragm area. And the peritoneal tissues, which are part of, which is mesentery and peritoneum, was one other group here, which we labeled metastatic group 1. And then metastatic group 2 is the lower half of the abdomen where we find the ovaries and the inguinal fat. And so to really investigate if the ECM is contributing to that preferential growth that we saw on the previous models, we looked at these groups and did Western blot to see if in the chemotherapy modulated environment, are we seeing increases of these components? And what we find is yes, these components are upregulated in this chemotherapy setting, and we are also seeing activation of alpha SMA, which is a marker of fibroblast activation. With this, we wanted to go ahead and have that in vitro system to assay either survival, adhesion, and other mechanisms, phenotypes for these cancer cells. So for this fibroblast cell-derived matrix system, we took NIH2-3 fibroblasts. We either given a vehicle treatment or a chemotherapy treatment. The chemotherapy treatment here is an IC30 of cancer cells. And based on how the system's set up, they are grown at confluence. So the chemotherapy itself is not really inducing a significant amount of cell death in these 2-3 fibroblasts. And this was adapted from Edna Kuckerman at the Fox Chase Cancer Center, who pioneered in making this fibroblast cell-derived matrix, just without the influence of different therapies that these patients may be going through. So what we can do is we can grow these fibroblast cell-derived matrices. We can then decellularize them and remove the cell body. So then we're just left with the extracellular matrix. And then from here, we can go ahead and plate cancer cells and ask a variety of phenotypic questions, such as survival and adhesion assays here. And when we look deeper into these fibroblasts, we can see that vehicle-treated fibroblasts versus these chemotherapy-treated fibroblasts become elongated, which is a sign of activation. As you can see here, they begin to elongate, and as I mentioned, this is a sign of activation for these fibroblasts. And in terms of these treatments, we are also seeing an upregulation of extracellular matrix. Yes.
Those are EMs of intact cells and then EMs of the matrix also.
Yes. So as my committee member mentioned, yes. So I'm showing you just the 2-3s, but we can decellularize them, and this is what they look like without the cell bodies. And without the cell bodies, as you can see, we can still see the matrix is still there. And when we look further, we can see this difference in the topography where due to the treatment, we are seeing that it's creating kind of more of this like rough topography in the chemotherapy treated FCDMs. So we asked the question, are we seeing, is this environment going to create an advantage for cancer cells to grow, especially like in low serum conditions or in stressful conditions? And we asked this question to show if this FCDM is really contributing to a survival advantage, then when we put them in low serum and they have to rely on the differences of this FCDM, will we see the survival advantage? And what we see is, yes, when we put these cancer cells in low serum and we put them in a chemotherapy modified FCDM, we do see that they have a higher advantage for viability compared to our vehicle FCDMs and also our no FCDM control. So this is just a traditional tissue cultured treated dish. And now to this model I was talking about. So the previous publication obviously coupled the cancer cells and the fibroblasts in the presence of chemotherapy. But we wanted to ask if we decoupled the cancer cells and the therapy, where the therapy itself modulate the in vivo environment for advantages of cancer cells, specifically in the context of our beta-3 negative and positive. So what we did here is we had a cohort of mice that were given vehicle or chemotherapy. And in our chemotherapy modified mice, we pre-incubated them with tweak. And here we did this because we know that in this post-chemotherapy setting, we have this enrichment of tweak. And in terms of the cells, they were isolated for either beta-3 positive and negative expression. And we isolated and made sure that they were of high yield because we did both magnetic cell sorting and also fluorescent activated cell sorting to have a very pure population of either beta-3 negative and beta-3 positive. And after that, we went ahead and injected these cancer cells in these conditions a couple of days after the last treatment of chemotherapy for the mice. And we injected 1,500 cells. So this is a very small amount of cancer cells. A lot of IP models usually use between 500,000 to 2 million cells. Here we used a very small amount to highlight if truly these beta-3 positive cells and tweak in this chemotherapy modified environment is really important, then a small amount of cancer cells, almost an untraceable amount of cancer cells, should provide the ability for aggressive tumors when you have beta-3 expression. And so what we found is that in the mice that are beta-3 positive and pretreated with tweak in that chemotherapy modified environment, we see the worst probability of survival, followed by the green line, which are just our beta-3 positive cells, then followed by our beta-3 negative cells, but with the tweak stimulation. And lastly, the ones that had the best probability of survival were the beta-3 negative cells, meaning that they were the least aggressive cells in these environments. And on the left, I'm showing you IVUS imaging of these mice throughout the time. And as you can see, these chemotherapy alter model with our AV beta-3 positive cells gave us the most aggressive tumors here. We wanted to measure that. So what we did is we did a high tumor burden measurement and this is mice that reached a luminescent counts greater to or equal 1e to the 7, which is a very high magnitude compared to our survival cutoff for 5e to the 5 luminescent counts. And as we can see here, this highlights what we kind of see in these images already, which is that this beta-3 positive tweak stimulated cells form the biggest tumors or the most aggressive tumors compared to our beta-3 positive or even those with tweak. And when we look at the different organs and where these cells would go to, we notice that in the mesentery, in those beta-3 tweak stimulated cells, we see this increased growth towards that tissue compared to others. And for AIM2 findings, we see that these peritoneal tissues from chemotherapy-treated mice had this enrichment of these ECM components that I was talking about, including activation of fibroblasts, and that this alters relevant ECM components that are implicated in these cancer stem-like cell survival, and that these beta-3 tweak-treated cells are more tumorigenic and show increased metastasis to the mesentery in a chemotherapy-modified mouse. And so the relevance in the field here, this paper focused a lot on beta-1 dependent manner and beta-1 dependent mechanisms. But here we want to highlight that beta-3 positive cells in this chemotherapy altered environment also have a predilection for growth in the mesenteric tumors, as we also observed in our other IP models where there was this preferential growth of cancer cells to the mesentery. And last aim, so we're almost done. So for my aim 3 was to determine if RelB mediated AV beta-3 expression can be targeted to reduce relapse potential. So the main goal here is to clarify the roles of NF-kappa B in inducing AV beta-3 during chemotherapy for survival of these cancer stem-like cells. So just to remind everyone, what we've seen is that when we knock down RelB here, we seem to induce the loss of beta-3 and to some extent also alpha V. However, alpha V beta-5, which is also important in this post-relapse environment, as I showed in that first aim, where after chemotherapy, you have this shift from AV beta-3, AV beta-5 negative to AV beta-5 positive population, but it was not NF-kappa B dependent. So we also have to find a way to try to block this AV beta-5 because we know it's important post-chemotherapy. And so we're going to do an IP model to do this. And now to highlight how we're going to block specifically AV beta-3 and AV beta-5 on the surface, because again, beta-3 seems to be partially regulated by RelB and NF-kappa B. However, AV beta 5 is not. We have to find a way to block that therapeutically. So I'm going to summarize a whole aim that I didn't talk about, which I pretty much focus on the adhesion aspect of these integrins. I kept talking about how they do adhesion mediated things. So I'm going to highlight this aim with 2 bullet points. So ultimately what we find when it comes to the adhesion here and rel A and rel B, loss of rel A and rel B in spheroids and also alpha 5 beta 1 antibody neutralization reduce adhesion to fibronectin in vitro, but not AV beta 3. And AV beta 3 and AV beta 5 integrants interact with vitronectin, and we want to block this type of interaction. And so in vitro, we use selingitide, and selingitide reduce the attachment of cancer cells to vitronectin. And this is what this molecule looks like. So now for this mouse model, we will be administering this drug to block that interaction with AV beta 5, AV beta 3, and vitronectin. So the way that we did this was we did another IP model with IVUS imaging, and we took our OVCAR8 RFP luciferase cells now with an inducible small hairpin RNA, either to a negative control, so that's what this SH-neg here is, and also to either target and reduce the expression of rel-A or also rel-B. And so what we did here is we went ahead and injected 300,000 cells in these mice, and after 7 days, we began to give them treatment. So in the SH-neg groups, we had a vehicle control, so these cancer cells were not given chemotherapy. In this other cohort, they were given chemotherapy. And if you notice, for the knockdown of rel-A and rel-B, we specifically focus on only the chemotherapy setting. We have previously shown what knockdown of rel-A and rel-B does without the presence of chemotherapy. But for this study, we really wanted to specifically focus on the role of rel-A and rel-B during chemotherapy. So that is why here, when we gave them chemotherapy, on that first treatment of chemotherapy, we switched the chow of the mice to doxycycline chow. This doxycycline molecule, these mice eat it on their food, and this induces that knockdown. So here we are able to see what knockdown of rel-A and rel-B does during the administration of chemotherapy. And after those treatments, we either put these mice on either vehicle or that selingitide maintenance therapy throughout the study. And ultimately, the way that we assayed and called endpoints here was when each of these cohort, when their average luminescent counts reach 5 to the 5 luminescent counts, that is what we considered endpoint measurements. And so we waited over time to see all these cohorts until they reach that luminescent average reading. And so what we found here, I'm showing you probability of survival. Right now, we're only looking at the SH-neg cells. And what we can see here, the black line, this is our vehicle. All mice quickly die, and their probability of survival is very grim. When we just administer selingitide alone, we see a small but not significant, very marginal difference in terms of probability of survival. When we give them chemotherapy, we do extend the life of these mice, as seen here in this red bar here. When you do chemotherapy and selingitide maintenance therapy, there seems to be a small change, but nothing very significant. When we look at rel-A, so rel-A alone in the presence of chemotherapy gave us a survival advantage here, and knockdown of rel-A with selingitide abrogated that. And we looked deeper into these studies, and because of time, I can't show you, but ultimately what we found here is that in terms of chemosensitivity, when we knocked down rel-A, those conditions gave us the most chemosensitivity. So essentially, when we knocked down rel-A, only, not with selingitide, we see a very significant drop in in terms of how many cancer cells died because they were sensitized to the chemotherapy. When we look at the rel-B knockdowns, what we can see here in the light pink, which is just knockdown of rel-B in the presence of chemotherapy, it's pretty much the same as just chemotherapy alone. So knockdown of rel-B in the presence of chemotherapy did not provide a survival advantage. However, when we couple that with the selingitide maintenance therapy, this last bar here, we do see that increases the probability of survival for these mice. And when we looked further into these tumors and look at our IVUS imaging, we noticed that we attribute this probability of survival for these knockdown of RelB with chemo and selengitide to the fact that when you look over time, that group over time had the least tumor burden for the longest period of time before um, eventually relapsing and reaching that 5 to the 5 count. And in terms of where the cancer cells are going, in terms of the different organs, we again did this assay, right, where we dissected the organs and in an organ-specific manner, we looked at the luminescence readings, ultimately leading to the tumor burden of each of these organs. And what we found was in the omentum, when we compared just the chemotherapy and we look at RelB with chemotherapy, remember that didn't really have much of a difference there. And what we see is that omental burden continues to be really high in these conditions. However, when we knock down RelB with chemotherapy, but also gave it selengitide, we see this drastic drop here, which we also attribute to this group living the longest. And ultimately, if we just focus on these two bars and we look now at the mesentery, the tumor load here is very high. And when we look at the mesentery, ultimately these mice succumb to the cancer due to this high omental burden. But however, for these cancer cells here, where the omental burden was low, ultimately when these cancer cells did relapse, they relapse and the growth was mostly on the mesentery side here. And finally, we wanted to look at what's going on in these actual cancer cells. So, and again, we wanted to focus on the omentum and the mesentery, where we saw most of our findings. So what we did here is we took these cancer cells from both of these sites and we digested them, and then we stained these cancer cells from these suspensions with an AV beta 3 antibody, as well as RelA and RelB, which are part of the classical and alternative NF-kappa B pathway, and as well as a nuclear stain. And we did imaging flow cytometry. So this imaging flow cytometry allows us to actually look at each of these cancer cells that were in these mice, and we can look at their nuclear stain, and we can look at RelA and RelB, and we can also see if there's colocalization of these factors into the nucleus, which will tell us that they are actively being used there, as you can see in these images here. So what do we see when we look at just the beta 3 positive cells and we are looking at either RelA or RelB, and this is nuclear, so this is the active factors. Off the bat, what we can really see is when we compare omentum and the mesentery, in the mesentery, RelB is really active in this tissue. And when we knock down RelB specifically with that selengitide maintenance therapy, we really see this drop of RelB activity in that tissue. When it comes to RelA, they don't really, these AV beta 3 positive cells aren't really relying on this RelA factor. And when we look at the beta 3 negative cells, we can see this drastic difference where in the mesentery, these beta 3 negative cells are not relying on RelB here. And ultimately, when it comes to total AV beta 3 expression, what we find is that AV beta 3 is, we can see this knockdown of this loss of AV beta 3 when we knock down RelB in the presence here. So with these findings, what we find that specifically in the mesentery, we are seeing this active RelB expression. And when we inhibit this, we see loss of AV beta 3, but only when combined with selengitide to inhibit AV beta 5 as well. And this interaction with vitronectin, we see the best survival outcomes here. And in terms of the relevance in the field, this puts into the idea that inhibiting RelB-dependent mechanisms, including integrin antagonists, that's important with beta 3 expression, may be clinically relevant as beta 3 has been shown to be important in many other cancer types. And we should also put high-grade serous ovarian cancer in this category for potential use. And for my final conclusion, and this to just bring it all together, I mentioned this idea of plants and trying to get rid of weeds, and how we can think of a tumor as a weed where the bulk of the tumor cell is like the top of the plant that you can see, while these stress tolerant cells express some of these markers like beta 3 and CD117 are like the roots. When we give it chemotherapy, it's like if you are just manually ripping up the top of the plant, but you're leaving the roots behind. And when you leave the roots behind, the tumor can come back or the weed comes back. And so ultimately here in the context of NF-kappa B and this endocrine signaling, what we can see is that in terms of the bulk of the plant, REL-A seems to be more important in this, while REL-B seems to be more important in terms of the roots and also AV beta 3 modulation. And with that, I'd like to thank Dr. Carrie House for all these years of working for her. Also, Michaela Robinson, the rest of the lab, my committee members for being here. And also a lot of alumni that I've known throughout the years. Dr. Ingrid at the microscopy core, Michaela again in the flow core. Special thanks to also Larkin at the vivarium. My industry mentor, Dr. Juan Cueva. Also our collaborators like Dr. Cherish, all of my funding sources, all of my friends and family as well. And yeah, it's been a journey. So with that, I can take questions. Including Zoom as well. I can answer questions on Zoom. Yes.
Do you see a relapse with cancer-like stem cells or cancerous stem cell-like cells? Do you see a difference or have there been any literature of the balance between tumor cells and cancer stem-like cells? Are there more of them after the fact?
Yeah, so the question was the proportion of cancer stem-like cells either before and after chemotherapy. So before chemotherapy, the cancer stem-like cells are a small proportion of the overall tumor bulk, so usually between 1 to 10% of the cancer cells. And after chemotherapy, we do see an enrichment where that ratio goes from predominantly the bulk of the tumor cells to predominantly cancer stem-like cells. Yeah. No, yes.
Do you have any kind of idea, say, you can treat the cells with tweak and these receptors come up. How long does that stay?
That's a good question. So when it comes to beta 3, it's an interesting one. So it seems that it's either enriched or induced in very specific situations, right? Like very stressful conditions. But then as you can see with Jenny's data, in early establishment, beta 3 seems to be upregulated and needed there. But once there's an established tumor that either the expression goes down or the population of those cells expressing that seem to be lessened. And it's been shown in multiple models that, especially when it comes to when you look at the primary site versus metastatic, it seems that this beta 3 expression is more important in metastatic sites versus the original tumor sites, and therapies exacerbate that.
If we ever looked at, I think what you're asking is if you stimulate with tweet and then 2 days later, 3 days
I was
later.
thinking about the assay, was stimulating the cells
With the tweak?
letting them go out.
Mm.
The chemotherapy.
Yes. So I guess another aspect of that is so yeah, we incubated with tweak for 2 hours, and this is from previous work that we know that after 2 hours of this incubation, we see this activity of LB. But also what's interesting in terms of the tweak story is that we looked at these different organs. So besides the extracellular matrix components, we also did an ELISA and we looked at tweak specifically on these different organs. And in the omentum, we actually see the most amount of tweak versus in our mesentery and our other metastatic site, the ovary and the inguinal fat, the tweak expression is low and the omentum expression is pretty high. So we think that maybe, and as we know, the omentum is the preferred site for these cancer cells. So we gave them this quote unquote artificial boost as to model what we've seen before, but we also know that the primary site also has the highest amount of tweak expression or tweak there. So I think that also continues to modulate that. And also when we look at the omental site, especially with AV beta 3 expression, it was continuously high. And even when we knocked down rho B in the omentum, we didn't see that drop as we saw in the mesentery. And we think that maybe this known enrichment of tweak in that organ specific like omentum may be modulating that and potentially a reason that we're not seeing the loss of AV beta 3 in that tissue compared to the mesentery. And also, we use cylindrotide, which has been used extensively. It's failed in the clinic. But the interesting thing about using cylindrotide, especially when it comes to these mouse models, is that there is evidence to show that cylindrotide itself in vivo models, and this is in the context of an immune competent mice. So that's another limitation to this study is that there is the rest of the immune system's not there. But in these models with cylindrotide, they see that cylindrotide itself is modulating macrophage polarization. And so there might be some of that also occurring in these models. And we do have ideas to do a macrophage panel to see if the cylindrotide in the omentum and the mesentery is being modulated differently to also account for some of those differences that we're seeing in those two tissues.
And also for the matrix production.
And for the matrix production. Yeah. Still no questions.
Any more audience questions?
Thank you, friends.
Don't have
Thank
any
you. Thank you.
In your model, CM, chemotherapy, all the
Mhm.
positions, CM atom, and like RNA interactions.
Are there other interactions there?
Yeah. CM.
Yeah, so the question was, in the chemotherapy setting, besides the ECM modulations, is there other?
So basically, does chemotherapy work in CM? As I see, you decellularize.
Yes,
CM.
I see what you mean. Well, yeah, without the cell bodies, we just have this kind of mesh of ECM proteins. Um, Ultimately, I mean, we do have a control, which is just a vehicle, and then we also have a non-ECM control. And so ultimately, the difference that we're assaying there is just the ECM versus I'm sure that we potentially may have different like phenotypes that we see if the cell bodies were there, because obviously the cancer cell, I mean the fibroblasts, they're not only just secreting the ECM, but they're also expressing cytokines and other soluble factors that in turn, when you think of the ECM and all those soluble factors is what we call the matrixome. So that's what that other paper really highlighted. Um, but for my context, is that I really wanted to just specifically isolate the ECM and see the ECM contributions, knowing that obviously there's other contributions, but like soluble factor contributions, but really want to just focus on that because at the end of the day, the extracellular matrix gets modulated and all these cytokines and all these things are always being modulated and concentrated different, but the extracellular matrix, it's kind of like the work that's left behind by these workers. So it's easier to drive some conclusions from that than having a system where I'm also have the cell bodies there. But also ultimately, who knows to what extent that matrix itself is contributing to these cancer cells. Although we know that all we need is a small contribution from this 1% of these cancer stem-like cells, and just a couple of them need to be in the right niche, right? So here's just highlighting that if you're in the right niche and you have this beta 3 expression, you may potentially be able to regenerate a tumor much quicker and faster than without these modulated extracellular matrix regions.
Dylan, were you asking if chemo affects the ECM proteins themselves, not just the cells that
But
produce them? Yeah.
But yes, the data I was showing you was that
Yeah, I think he's just asking not just the production, but pre-existing ECM. Does chemo affect that?
oh Okay, there's two ways. So the fibroblasts are actively modulating and changing those environments during chemotherapy, right? So that is happening. And so even in this system here, before I add the treatment, the first step is to plate these 3 to 3 NIH fibroblasts at a high confluency. So meaning by the next day that I actually add the chemotherapy, they already have a good matrix made. And then we give them the chemotherapy in that context. But just to add more into like a more broader term is besides the fibroblasts, making this extracellular matrix. We also know like cancer cells themselves make their own ECM. And I didn't highlight that with the Go analysis, but if you look deeper into the Go analysis, there's some things there with extracellular matrix organization. And when you go deeper into that extracellular matrix organization, beta 3 does pop up in in those set of genes. So besides also, so it seems that especially so that's a little bit more important in terms of the context of cells that are just traveling around the peritoneal cavity. So it's a very dynamic process, and the RNA-seq data is really cool because you get to see cells when they're attached to something versus cells when they're floating, and how rel A and rel B is important in that process. And also, just to complicate things more, macrophages also work with fibroblasts to modulate extracellular matrix. And extracellular matrix and the production and structure of it can also polarize macrophages from one side to another to just make things even more complicated. I hope I didn't ramble too much about that, but yeah. I did, yeah, yeah.
Any other questions?
What's next, Omar?
Submission of this manuscript to Nature Communications, and I'm actively looking for roles. So if anyone knows, please let me know. And that's kind of it. I mean, I have the summer to kind of figure that all out. So submission, wait for reviewer comment, see what's going on there, and also actively looking for roles. Preferably, I like the industry, but due to the current administration, things are looking pretty tough, because they don't want us to know the truth, and the truth is a gateway to science. But anyways, um, yeah. So the next big things are submission of this manuscript, see it through, help in that process, continue to look for roles, and leaning towards industry roles, but I'm also open to postdocs as well. It really is a combination of um, financials and also the science. So don't mind taking less money if I think the science is really cool. And yeah, so that's kind of where I'm at right now.
How about some R&R here?
Yeah.
Yeah, that's my question.
Oh yeah.
That's it.
This was taken years and years ago in Maui. This is at Black Sands Beach on the way to Hana. Yeah. I always like to just end it because it's like, oh, it's a nice thing to see at the end of a presentation. Yeah.
If there aren't any other questions, we're gonna take aside to this topic. And we'll have some time with the big committee.
Thank you everyone on Zoom for joining. I hope you enjoyed the presentation. Love you all. Thank you. And bye.